Hello, my name is Sarah Maines, and I'm the Liver Cancers Program Director at Global Liver Institute, the world's largest liver patient organization. And at GLI, we bring together more than 200 organizations across our councils, campaigns, and events to advance liver health as public health priority. Our mission is to prevent liver diseases, improve early detection, and scale the most effective strategies for supporting individuals and families impacted by liver disease and liver cancers. In this first episode of our October is for Livers Liver Cancer Campaign Series, we explore new frontiers in T cell therapies, a promising area for patients with advanced or recurrent liver cancer. As we continue to face limitations with surgery, chemotherapy, and current immunotherapies, there is a growing need for novel, more effective treatment approaches. We're pleased to welcome today Dr. Stephen Katz, who will guide us through the evolution of immunotherapy, the role that the immune system and tumor microenvironment plays, and how T cell therapies are being developed to improve patient outcomes. He'll also address recent clinical trial data, ongoing challenges, and what's ahead in this emerging field. Dr. Katz, thank you so much for being here with us today. I'll turn it over to you to present now.
Well, thank you, Sarah, for that wonderful introduction. It's always great to be with my friends from GLI, and thank you for the opportunity to talk about what I think is a very important and I think a very interesting and exciting topic. We're going to cover new frontiers in T cell therapies for liver cancer, and we're going to be delving into the realm of immunotherapy, which has arguably given us one of the greatest sets of advances in oncology and medicine in general over the past few decades. But unfortunately, this has impacted some types of cancers more than others. And I think we can all agree that for patients with liver cancer, and we'll discuss the different types, we have more work to do. And so we're going to talk about recent advances, hope and promise using cell therapy, which is a form of immunotherapy, some of the challenges that face us when we're developing new treatments for the liver in particular. And hopefully you will all agree that this is a very exciting and promising frontier for these patients who desperately need new therapeutic options. And so, as I mentioned, we'll cover a little bit about immunotherapy in general, where there has been some very important success for patients with liver cancer, but where some of the challenges remain. We'll get into what cell therapy is and what the advantages may be in applying it to patients with liver cancer and what the potential impact for these patient populations could in fact be if it realizes its full potential. So I think it's helpful to spend a couple of moments defining which patient populations we're going to be talking about. So liver cancer as a term or a concept or an idea encompasses a heterogeneous or broad array of different diseases. The most common type that we often think about is primary liver cancer, with hepatocellular carcinoma being at the top of the list. In second place, a distant second, is cholangiocarcinoma in terms of incidence, which is bile duct cancer that can arise within the liver itself. And then we have patients who develop secondary or metastatic liver cancer, which is when cancer starts somewhere else in the body, most commonly the colon or the rectum, colorectal cancer, and then spreads to the liver. Now, these are different diseases with different issues facing these patients, but there are some common themes in particular that make immunotherapy challenging. And so we'll get into that when we talk about the different types of immunotherapy and cell therapy in particular. And so one of the questions that we often think about as physicians is, why is the liver such a common site for malignancy or cancer in the first place? Part of it has to do with the nature of the distribution of blood flow in our bodies, and this may apply more to metastases, secondary liver cancer. All of the blood, essentially all of the blood from the colon or rectum, for example, flows through the liver. So it's not shocking in that sense that a lot of the cells that break off from the primary tumor end up in the liver forming tumors or metastases. And that's probably an oversimplification and certainly not the complete story, but it may be part of the explanation for why so many patients develop tumors in the liver. A big reason why patients develop primary liver cancer is the fact that hepatocellular carcinoma and cholangiocarcinoma are very often associated with conditions linked to chronic inflammation. whether it's hepatitis B infection or hepatitis C infection or metabolic associated syndromes. When the liver is subjected to chronic inflammation, repeated cycles of cellular injury and healing and cellular proliferation, there is a greater opportunity for mutations to develop, for cellular programming to go awry, and hence for malignant cells to emerge and tumors or cancer to develop. So chronic inflammation is a big reason why cancer develops in general, but in the liver in particular. And then third on this list, which is perhaps most germane or relevant to what we're going to be talking about today, immunosuppression. That is the liver's very powerful ability to shut the immune system off. And we'll talk about what the mechanisms for this are, why it's actually something that's helpful for daily existence, but unfortunately, why it's such a big problem, not only in terms of the development of cancer, but also when we're trying to apply immunotherapy for these patients. Immunosuppression in the liver does make it significantly more challenging, but we think there are emerging solutions in general, and with cell therapy in particular, that may give us the ability to overcome this. And so what are the current therapies and their limitations when we think about treating liver cancer patients? There's no question that collectively we've made tremendous progress in treating liver cancer patients, both primary and metastatic, with standard of care therapies. But I think we can all agree that there are limitations, and the vast majority of patients who need curative options or long-term disease control aren't getting that yet. They're not getting that today. But let's talk about each of these and what the limitations are or may be. So surgical resection, surgically removing a tumor, is a potentially curative option, like liver transplantation, but it's limited in the sense that many patients come to physicians with too many tumors or too high of a disease burden, or maybe the tumors have spread beyond the liver to the point where surgery isn't a viable option. And the same applies to liver transplantation, in addition to the issue of there being an insufficient supply of organs. We have TACE and TARE, which is chemoembolization and radioembolization. These treatments can provide very meaningful disease control in the right patients, but again, far too many patients come to us with disease too advanced, or liver inflammation or cirrhosis too advanced to benefit from these treatment options. So they're somewhat limited in terms of the patient populations that they could be successfully applied to. And then when we think about the patients that come with or present with or develop disease that's more advanced or has spread beyond the liver, then we start to think about systemic therapy, which is therapy given throughout the entire body. We think about chemotherapy, drugs like doxorubicin or cisplatin, or small molecule inhibitors, drugs like sorafenib. These drugs have unquestionably offered benefit to liver cancer patients, but they lack, in most cases, a durable long-term benefit or a durable impact on the patient. And that's where immunotherapy may offer more promise, because when you treat somebody with immunotherapy, if you think about it like a vaccine, the goal and what we're seeing in certain cancer types is that when we have success with immunotherapy, the patients can benefit from long-term effects of the drug, so very long-term and durable disease control, even to the point where we use the word cure in certain cancer types. And we've seen some success in liver cancer patients for sure with checkpoint inhibitors, a certain type of immunotherapy that I'll get into in more detail. But as I noted at the beginning, not enough patients are benefiting from it, and immunosuppression in the liver is a big reason why. And so we'll talk about that and how cell therapy may overcome that. And so I think it's always helpful to take a step back and talk about some of the basics of how our immune systems work, because that really sets the table to understand what we're trying to accomplish with immunotherapy and how immunosuppression may limit the ability of immunotherapy to work in liver cancer patients. So the immune system can be thought about very simply as being comprised of two major arms or components. One is the innate immune system, and the other is the adaptive immune system. So when we are faced with an infection, cancer, liver cancer, we would like our innate immune cells, things like dendritic cells and macrophages, to recognize the abnormal proteins, such as mutated tumor antigens, as something that shouldn't be there, a danger signal, and then take those proteins and then present them or show them to T cells, part of our adaptive immune system. and the goal being to develop an effective cellular or T cell response, CD4 and CD8 T cells that can recognize the cancer or the tumor and then destroy it by engaging it directly through recognition of those antigens. And a secondary goal, and a very important goal, which I alluded to earlier, is the ability of our immune system to demonstrate memory. Many of you will be familiar with this concept from the world of vaccines against COVID or the flu and pneumococcal pneumonia and so on and so forth, but it's also possible for our immune systems to develop memory T cells that can recognize tumor antigens. And if this can happen, this is the scenario in which patients develop long-lasting protection against their malignancy. And so this is our goal. But unfortunately, cancer in general and the liver in particular are very good at shutting the immune system off and suppressing immune responses. And that's what this slide is meant to illustrate, that there are things called checkpoint molecules like PD-1 and TIM-3 and LAG-3 that normally function to control our immune responses. And this is typically a good thing, because when we develop an immune response, a T cell response, again, using these examples to COVID or the flu, we want that response to be effective, but we want it to be time limited and limited in scope, because if the immune response is too exuberant or too strong or lasts too long, we can get sick and develop inflammatory injury to our normal tissues or autoimmune disease. And so PD-1 and other checkpoint molecules actually can turn the immune system off when it's done its job. The problem in patients with liver cancer and other cancer types is that cancer cells can actually hijack this checkpoint or control system that our T cells normally benefit from. And so cancer cells can actually express the ligand for PD-1, PD-L1 that can bind to PD-1, and that inappropriately turns T cells within the tumor microenvironment off. And beyond that, cancer, and liver cancer in particular, has an abundance typically of immunosuppressive cell types, regulatory T cells or Treg, MDSC or myeloid-derived suppressor cells, which are particularly problematic in the liver, that can also shut T cells off. And thinking forward to the later stages of this presentation, can also shut down immunotherapy and shut CAR T cell or T cell therapy off. So when we think about developing new therapies and immunotherapy in particular for patients with liver cancer, it's very important to understand how big of a problem immunosuppression is and what the mechanisms are, so that we can rationally develop therapies capable of resisting this and even reversing it. And so let's spend a moment looking at some of the currently approved immunotherapies for primary liver cancer, hepatocellular carcinoma, or HCC, how they work and where the limitations may be coming in. So drugs like atezolizumab and durvalumab, so these are checkpoint inhibitors, and they're blocking the interaction that I've circled in purple in this image. This is PD-1 binding to PD-L1. So these antibody therapeutics can block this interaction, so they're preventing a negative signal from impacting the T cells. So this allows the T cells within the tumor microenvironment to become reinvigorated or reactivated, and then they can recognize and attack the cancer. This has resulted in some very impressive clinical outcomes in HCC, although the majority of the patients still do not benefit from this type of therapy, even though it's a remarkable advance. And many believe that immunosuppression within the liver is a big reason why not enough patients are benefiting. But also, as you're seeing from the complexity of this diagram, these are very redundant pathways, meaning there are many pathways or molecules that do the same or similar things. So blocking just one of these checkpoint interactions leaves many open to exert their negative influence on T cell function. And so when we're developing therapeutics, the complexity of this immunosuppression should be contemplated as well. And finally, it's worth mentioning that for checkpoint inhibitors to work, you do need to have a certain level of T cell presence in the tumor microenvironment, because these drugs, unlike traditional cancer therapy, aren't targeting the cancer cells. They're actually targeting the patient's own immune cells. And so if the immune cells aren't present in the tumor microenvironment, well, there's effectively no target for the drug. And unfortunately, many types of tumors within the liver are what we call immune excluded, meaning it's difficult or impossible for the T cells to penetrate and exist within the hostile tumor microenvironment. So this is another point where cell therapy may be helpful because, as I'll tell you shortly, we can effectively manufacture a T cell response to the cancer and then directly administer it to the patient and even within the tumors in the liver. And so when we think about why the liver is such a difficult problem for patients getting immunotherapy, uh, we look to the data as always. And this is some of the most compelling and convincing data that the liver is, in fact, a significant barrier to immunotherapy success. So these are 3 separate studies looking at patients with metastatic disease, so cancer that's spread throughout their body, melanoma, lung cancer, and other solid tumors. And each of these studies broke the patients down into 2 groups, those who had liver metastases and those who did not. So these are patients with tumors throughout their body with or without liver metastases. And looking at these survival curves, what's very clear is that the mere presence of liver metastases, which is the bottom curve on each of these 3 survival graphs, the mere presence of those liver metastases was a significant predictor of immunotherapy failure, not just for tumors growing in the liver, but throughout the patient's body. So there is indeed something profoundly immunosuppressive about the liver that's not only allowing tumors to grow in an uncontrolled fashion, but also limiting or preventing the ability of immunotherapy to work in these patients. So we have to think very carefully about how immunosuppression is operating in the liver and develop therapeutics that can address this biologic problem. And cell therapy, I believe, and others believe, gives us an opportunity to finally tailor our treatments at a genetic and molecular level to hopefully address some of these issues. And so a word about why the liver is so immunosuppressive. Why do we want that to be the case for daily existence? Because it's not something that's only a negative for cancer patients. It's actually something that we benefit from day to day. And the reason is fairly simple. Everything we eat, for the most part, passes through our liver. The portal vein connects our gut to the liver, so the blood flows in that direction. And a lot of foreign antigens that we take in that our immune systems may otherwise react to strongly are passing through the liver. Also, our gut, as you all are familiar with, is full of bacteria and bacterial cell wall products, things like endotoxin or lipopolysaccharide. And so one could imagine that if the liver immune system was very responsive, this would create a very challenging situation for daily existence. So immunosuppression or tolerance is sort of the norm in the liver, but this, of course, is very problematic for patients with primary or metastatic liver cancer. And these mechanisms of immune tolerance, as I showed you and explained earlier, can be hijacked by malignancy or cancer cells to turn the immune system off and also limit the effectiveness of immunotherapy. But the immune tolerance in the liver and the immunosuppression of the liver has been well documented in many, many publications. And I'm just showing you a few on this slide. And immune cells in the liver, whether you look at T cells or dendritic cells or endothelial cells and so on and so forth, all tend to take on very suppressive or tolerogenic properties within this organ. And this is explained even at a molecular level. When we look at myeloid-derived suppressor cells, or MDSC, which I mentioned earlier, and we look how they function in the liver specifically, comparing them to lung MDSC, for example, what we've learned is that liver MDSC have a very specific pathway that's driving their survival, their expansion, and their suppressive function. This is driven mainly by a molecule called STAT3, which is a transcription factor. And the point I'm making here is that there's a very specific, even molecular biology in the liver that's driving immunosuppression. The good news to understanding this and attaining this knowledge is that this does give us very specific and rational therapeutic targets to potentially undo immunosuppression in the liver. And this may be achieved through the use of things like toll-like receptor 9 agonists or other innate immune stimulators, or cytokines that can activate the immune system. Things like IL-15 and IL-21, which I'll show you later, can actually be built into cell therapy products and then secreted to help support that type of immunotherapy's function within the liver. And so having covered that, and we've developed a bit of an understanding of why the liver is so challenging from a clinical and an immunologic perspective, let's talk about cell therapy, what it is, and how we can leverage this powerful therapeutic platform to help more patients with primary and metastatic liver cancer. And so, what's the reason for even thinking about cell therapy? Why do we want to do it in the first place? So, if we take a step back, what is the goal with immunotherapy in general? And one can answer that question probably in a few different ways, but I would say that one of the primary goals of immunotherapy is to provide the patient with a tumor antigen specific T cell response, sort of like what we're trying to achieve with the COVID vaccine. We want the patient to develop T cells that can recognize infected cells and eliminate them. Well, with cancer immunotherapy, it's very similar. We want our patients to develop very strong, robust, and long lasting T cell responses specific for their cancer cells. And so there are several ways to go about this. Vaccines, as we've done with infectious diseases, is one approach where you inject or infuse the patient with tumor antigens, mutated tumor proteins that can then be recognized by the immune system, as I showed you in that basic immunology slide, and then the innate immune cells stimulate the T cells to recognize the cancer, and so on and so forth. This is attractive in theory, but it's challenging clinically because that immunosuppression that I've been describing limits the ability of patients to respond to vaccines. That doesn't mean vaccines haven't worked or can't work. Likely there's going to be a role for vaccines, particularly in combination with other types of immunotherapy, but the immunosuppression is a big limitation. Checkpoint inhibition, which we've covered, these are the approved drugs for hepatocellular carcinoma that I mentioned earlier. They work very well in a certain percentage of patients, but again, the immunosuppression is limiting. And maybe more importantly for this type of therapy, the lack of a sufficient T cell population in these immune excluded tumors makes it difficult for checkpoint inhibitors to work in most patients because, again, with these drugs, we are not targeting the cancer, we're targeting the patient's T cells. And so you need to have a robust T cell infiltrate within the tumor microenvironment for these types of drugs to work. So that is a limitation. Adoptive cell therapy offers the opportunity to manufacture a T cell response specific for a tumor type or tumor antigen in a manufacturing facility, and then infuse that or give that back to the patient. And so the T cell response can actually be generated ex vivo outside the patient and then given back, which in theory bypasses some of these limitations that I mentioned earlier. There is still the problem of immunosuppression. There are other challenges with cell therapy that we'll get into, but that's the attractiveness of cell therapy in principle, is that you can literally manufacture an immune response specific for a type of cancer and give it to the patient. And we've seen unbelievable, remarkable outcomes in certain disease types like lymphoma and leukemia. And this is where most of the FDA approvals for cell therapy currently lie. Solid tumors are more complicated because of the tumor microenvironment, because of immunosuppression, because of drug delivery challenges, and so on. But we've seen enough evidence that cell therapy can work in cancer patients that we believe that we can engineer products to be effective in solid tumor patients and in liver cancer patients. And we'll speak about that hope and promise in a bit more detail in a subsequent slide. And so just to look at what a T cell therapeutic looks like, what is a CAR T cell? So there are many different or several different sources of cells that we can genetically modify and give back to the patient. And I'll get into that in a moment. But in principle, so we take T cells either from the patient or from an unrelated donor, and then we genetically modify them to express a molecule, a protein molecule that can recognize a tumor antigen. This can be a chimeric antigen receptor, which I'm showing you here, or a T cell receptor. But looking at a chimeric antigen receptor, or a CAR, and that's where the term CAR T cells comes from, you're taking a portion of an antibody that recognizes the tumor antigen, and then you're linking it to other molecules that can signal and activate the T cell. So this chimeric protein together can help the cell bind to the cancer, and then it provides a signal to the T cell, often 2 or 3 signals to activate the T cell, so then it can seek and destroy and ultimately kill the cancer cell. So it's this cycle of harvesting the cells, genetically modifying the cells, expanding them so that you have enough of a dose to give the patient, and giving it to the patient, where the T cells then have the opportunity to recognize the cancer specifically, hopefully, and kill the malignancy. And this is what it looks like in a laboratory. So this is an image from my lab showing a CAR T cell, which is the cell with the red halo. This is a CD3 stain, which is a T cell marker binding to a tumor cell and causing apoptosis or cell death. And the green staining on the side of the tumor cell facing the CAR T cell is an apoptosis marker called caspase-3. So when you bring the CAR T cell and the cancer cell together, this is what you'd like to have. This is what you get in principle or in theory. The problem for patients with solid tumors, it is just not this simple, because you have the immunosuppression, you have the issue of trafficking, meaning how do you get enough of your CAR T cells to actually make it to the liver? And then when they're in the liver, how do you get them to actually penetrate these very hostile, high-pressure tumor microenvironments? Then there are other issues outside of this molecular or cellular problem around cost and global scalability. These are very expensive treatments. The treatments that are approved today can cost several $100,000, and there's a very rational, value-based argument to make for those numbers. But I think we can all agree that's not scalable globally, or even for the majority of patients in the United States. And so we have to come up with ways of creating and delivering cell therapy products that are much more cost efficient and less expensive, so that many, many more patients can benefit from them. And there are certain types of cell therapy that give us an opportunity to lower the cost of goods and hence the treatment costs quite substantially. And so we need to think about that as well. It's not only about creating a therapy that's safe and effective. Obviously, we need both of those things to happen, but we also need to create therapies with the commercial model and the payer model in mind, so that these are viable opportunities for patients who need them across our country and across the globe. And so let's talk about trafficking or product delivery. So one of the issues that's fascinated me throughout my career and where I've spent quite a bit of time is drug delivery. And this applies to cell therapy products as it does to other types of therapy. It is very difficult to get a therapeutic to penetrate into a high-pressure liver tumor. Simply delivering a therapeutic, a CAR T cell, into a vein or a port doesn't necessarily mean you're going to get a sufficient or anywhere near sufficient quantity of that therapy into the liver or inside the liver tumor. So the way we deliver our therapeutics is quite important, probably something that has not received enough attention in drug development. But certainly for liver patients, we're aware of the benefits of regional delivery because we do that with chemoembolization and radioembolization. And so I would argue we should apply the same type of thinking to immunotherapy and T cell therapies as well. And so we'll talk about this, and I'll show you some data that illustrates the potential benefit of taking innovative delivery approaches with immunotherapy. And so this is some of that data. This is a phase 1 trial looking at a CAR T cell product targeting an antigen called CEA for patients with colon cancer and pancreatic cancer, liver metastases. And in this trial, in addition to looking at safety and responses, we looked at delivery. We looked at the number or frequency of CAR T cells in the liver tumors by doing biopsies or needle samples pre and post treatment. And we were giving the cells directly into the liver using a regional delivery approach like chemoembolization or radioembolization. But we were comparing two different delivery technologies. We were looking at a standard catheter and then using a method called pressure-enabled drug delivery, which enhances the pressure gradient and the flow to the tumor. And we saw that with the pressure-enabled approach, we were getting significantly more of the CAR T cells into the liver tumors, which resulted in some really nice responses in these patients with liver metastases. And on the lower left, I'm just showing you that in the blood, the duration of exposure was quite limited, only for about a week, where we're able to detect the cells by PCR, which in many patients is desirable, because if a patient has tumors only in the liver, not all do, but some patients will have tumors only in the liver, you don't necessarily want the CAR T cell spending a lot of time in other organs, because this could lead to off-target toxicities. The selectivity of getting the cell therapy product into the liver and keeping it in the liver, I think can be an advantage for HCC patients, cholangiocarcinoma patients, and certain patients with liver-dominant metastatic disease. And so there are different types of cell therapy products, and they all have pros and cons, advantages and disadvantages. So let's walk through some of the different types, and then we'll get into some of these in a bit more detail. And I think at the end of the day, in the future, we're likely going to see approvals of all of these different types. And I think that's going to be a great thing for patients. There'll be different options, either in sequence or even in combination at some point. And so let's go through them. So autologous really is the first type of CAR T-cell therapy to make significant progress and show promise. And the FDA approvals I mentioned are autologous products for the leukemia and lymphoma patients. And what this means is we're taking the patient's own T cells out of their bodies. We're re-engineering them with genetic modifications, mainly to express that CAR protein for a specific tumor antigen, and giving it back to the patient. The advantages are, well, there's no risk of immune rejection because we're giving the patient's own cells back to them. We have the most experience and the most data here in terms of feasibility and safety and effectiveness. The disadvantages, very expensive. There's variability from patient to patient because every time we make a product, it's essentially a unique product from the patient's own immune cells. So every batch, if you will, is going to be different. So there's variability every time the patient gets the product, which does add some unknowns into the treatment situation. There's a waiting time. So it can take about 4 weeks to go from vein to vein, meaning taking the cells out of the patient, manufacturing them, engineering them, and then giving them back. And for certain types of cancer, 4 weeks can be just too long to wait. And so that is one of the disadvantages of autologous therapy. And then we have a couple of types of allogeneic products, which means taking cells from an unrelated individual, like a liver transplant. You're taking an organ from someone who's unrelated and giving it to a patient. Here we're taking T cells, for example, from an unrelated person. We're modifying those cells to express a CAR protein and other immunologically active proteins, and then giving them back to the patient. The advantages are shorter waiting time. This is a so-called off-the-shelf proposition. The cost tends to be lower. There's still some variability when you're getting the cells from multiple donors. And you have the probability or the possibility of immune rejection. Like with a liver transplant, when you're giving somebody cells from an unrelated individual, the recipient's immune system can recognize those as foreign. And so that's a problem, and we need solutions for that. Inducible stem cells, or iPSC, is a newer form of allogeneic cell therapy where we can take adult cells, program them to de-differentiate it or go backwards, if you will, to become stem cells, and then we can differentiate them into different cell types like T cells. And so this gives us the ability to make, in theory, an unlimited number of doses from a single donor. So you eliminate that issue of variability, lowers the cost of goods. And this is something that's potentially much more scalable nationally and internationally to apply it for more patients. And you have that shorter waiting time. The disadvantages are it's technically more challenging, but progress is being made. It's the newest approach, so we have the least data, but this is something that's very promising. And then there's vivo editing, where you can use viruses or mRNA technology to modify the patient's own T cells. This is very early, and we're still learning a lot about how this could be applied to different types of cancers and liver cancers in particular. And so let's talk a bit more about iPSC, or inducible pluripotent stem cells. This was discovered in Japan. Dr. Shinya Yamanaka won the Nobel Prize for this in 2012 for discovering how to create iPSC, and many others, particularly in Japan, like Dr. Shin Kaneko, pictured below, have developed processes through which we can actually differentiate iPSC into T cells reliably. One of the other advantages of this type of immunotherapy is you can make a large number of genetic edits. So you're not just putting in the CAR to recognize the cancer, but you can put in different cytokines to stimulate the cells and to modulate the immunosuppressive tumor microenvironment. You can also do things to modify the cells to allow them to evade the patient's immune system. So you can avoid the immune rejection by giving these cells the ability to hide from the patient's T cells and natural killer cells. So using iPSC technology, the number of genetic edits that we can make in an individual product is larger, and that gives us the ability to make therapeutics with a much more robust array of immunologic functions, and gives us the ability to tailor the therapy in a very fine way to hopefully match the biology and the immunosuppression pathways that are present in the liver, for example. Now, anytime we develop a therapeutic, we have to think about safety, and the safety implications of cell therapy can be profound. Things like cytokine release syndrome, neurotoxicity, off-target effects, meaning recognition of the antigen or a similar antigen in normal tissue. So these are things we're very, very thoughtful about when developing cell therapy. We can do things like add suicide genes to eliminate the cells if necessary. We have drugs that we can give patients to treat some of these side effects. But these are, potentially very potent therapies. And so having plans in place to address the safety issues is very, very important. And of course, this is front and center with the development of any cell therapy product. And I always like to mention, I think it's important to mention that when thinking about immunotherapy for liver cancer, it's critical to look at how we're measuring response. I worry that we miss activity in immunotherapy trials at times because we're using conventional response criteria, which look at changes in tumor size. One of the problems with immunotherapy and measuring response is that tumors in the short term can at times actually get bigger with effective treatment, because when you cause an effective immune response, there's inflammation, you have cells rushing in, you have water leaking out of blood vessels, like when your shoulder gets sore from a vaccine shot. Well, that happens in tumors to a similar degree. And so you get tumor enlargement that's unrelated to the cancer cells dividing, and we call this pseudoprogression. It doesn't happen in every patient, but it does happen. And so when we measure response to immunotherapy, we should be thoughtful about how we do that. And here's an example from a patient I treated in a CAR T cell study, showing you at the first follow-up imaging, the tumor actually looked bigger on the MRI. It got brighter on the PET scan, but when we looked a few weeks later, we saw that there was actually shrinkage and loss of signal on the PET scan. And so a couple of lessons here. One, you want to give the patients time. If they're doing well clinically, it might make sense to keep them on therapy and continue to follow them because the response can evolve on the imaging studies over time. Using other readouts like circulating tumor DNA or liquid biopsies can be very, very helpful as well. And so moving on to the third and final section, let's talk about the potential impact in more detail for liver cancer patients. Here's an example from that trial I mentioned earlier when I showed you the CAR T cell delivery data. This is one of the patients with pancreatic cancer liver metastases who had a good response. And so by getting a very high number of the CAR T cells into the tumor through a very focused delivery approach, this patient did have a good response on imaging. The tumor markers came down, and the patient had a reasonably good survival time compared to benchmarks. Looking at this immunofluorescence set of images on the right, you can see that in the red and green, staining for T cells and the CAR protein, we're seeing a high density of the product in the tumor. So the delivery approach, I think, can be very helpful, and doing immunotherapy regionally for liver cancer patients in particular may be a very rational approach in certain settings. And then there's also been some very promising data out of Baylor looking at a specific tumor antigen, which is very commonly expressed in hepatocellular carcinoma patients. This antigen is called GPC3. These investigators infused GPC3 specific CAR T cells to these patients. These cells also express the cytokine IL-15, which can activate T cells and allow them to be more effective at killing cancer cells. And they had some very nice outcome data in this study. These anti-GPC3 CAR T cells were well tolerated, and they also saw some evidence on imaging where the initial images may not have shown responses, but eventually responses were noted. We also saw examples in this study of lesions that did not necessarily shrink on the scans, but they became non-viable, meaning the tumors looked dead on the imaging, even though the size didn't decrease. Again, reminding us that measuring response in immunotherapy requires perhaps a slightly different approach than we've traditionally taken in oncology drug development. And this is an example of a product that's going to be coming into the clinic in late 26, early 27 for patients with HCC and liver metastases. This is an iPS T cell product. This product is also going to target GPC3. Here it's using a T cell receptor instead of a CAR, which allows it to recognize the protein that's expressed intracellularly in the tumor when it's presented on the surface inside of a specific HLA type or HLA molecule. And then you can see all these other proteins that are being expressed, including cytokines, IL-15 and IL-21 to support the product function, but also to help modulate the immunosuppressive cells. And then these other edits are designed to do what's called immune evasion, which allows this product to hide from the patient's T cells, antibodies, and natural killer cells to hopefully prevent allorejection, which will ideally not only allow these cells to last longer in the patient and provide a more durable response, but also allow these cells to be dosed multiple times without the patient rejecting them. And giving multiple doses may be important in liver cancer cell therapy because these are difficult to treat tumors. Trying to achieve a complete effect or a complete response with a single dose, like we've seen in leukemia, may be very challenging, maybe too steep of a hill to climb. And so multiple doses may be required. And this is a summary of how these cytokines are intended to work. IL-15 and IL-21 can stimulate the cells to kill cancer, and they can also eliminate immunosuppressive cells. Again, one of the advantages of inducible pluripotent stem cell technology is the ability to make multiple genetic edits, giving your cell therapy product multiple immunologic functions, which may be needed to address the complexity of the tumor microenvironment and the immunosuppression within it. And a brief summary, I'm not going to go through each of these, but I just want to give you a sense or a flavor for how many cell therapy trials are currently ongoing or soon to be opened targeting GPC3, again, this tumor antigen that's very commonly expressed in hepatocellular carcinoma, but also colorectal cancer and other types. And so it's very promising and it's very encouraging to see this many companies moving into this space, trying to develop novel cell therapies for liver cancer patients. And the more companies that are willing to invest in this and go after these targets, the more future options we're likely to have for patients with primary and metastatic cancer in the liver. And so to summarize, checkpoint inhibitors have shown great promise and great efficacy in liver cancer patients, HCC in particular, but we need to find more ways for patients to benefit. Cell therapy offers the ability to manufacture an immune response, and in the case of iPSC, to do multiple genetic edits to create complex functionality tailored to the tumor microenvironment. Barriers to cell therapy, including immunosuppression and trafficking or delivery, may be overcome in the near future for the reasons I mentioned already. The delivery approaches we've discussed, it may make sense in liver cancer patients to give cell therapy products regionally, at least as an induction therapy, and then maybe systemic maintenance. But giving the cell therapy product regionally may give it a better chance to induce a meaningful clinical benefit. The response assessment strategies need to be thoughtful and tailored to the immunotherapy mechanism of action. And as I've noted, having these multifunctional cell therapy products or combinations of different types of immunotherapy may be where most of the hope and promise lie. And so with that, I think we have some time for questions.
Dr. Katz, thank you so much for such a great and impactful conversation today about immunotherapy and T cells. It's so helpful to dive in more and learn about the innovations in this space. I'd like to ask a couple questions and want to first start with, why is the liver such an immunosuppressive organ? I think a lot of viewers often have questions about that. And I'm wondering, how is this different from other organs?
Great question, and I've spent most of my career thinking about it and trying to help us answer it. And many people have made a lot of progress in getting to that answer. And again, I think we've evolved, if you will, to have very tolerogenic or immunosuppressive livers because so much of what's in our guts and what we eat passes the portal vein and the bloodstream that flows through the liver. And so we need that tolerance for daily existence. But what happens is the immune cells in the liver become conditioned to be so immunosuppressive that malignancy, primary liver cancer, metastatic liver cancer can hijack that and take advantage of it and prevent the patient from mounting an immune response to the cancer and also prevent immunotherapy from working effectively in these patients. And so when I say we need combinations of immunotherapies or that we need cell therapy products that have multiple functions, what I'm getting at is that we not only need to think about attacking and destroying the cancer cell, we also need to think about addressing the immunosuppression and turning these suppressive immune cells off. And so we need to achieve multiple things at once therapeutically within these complex, liver tumor microenvironments.
Thanks, Dr. Katz. Next question, how big of a problem is response assessment using imaging for immunotherapy in the liver? I'm wondering what we can do to address this problem to accelerate bringing new treatment options to the bedside. What would you say about that?
I think it's a very significant problem for immunotherapy in particular, but for liver therapeutic development in general. That's why we've developed things like modified RECIST or mRECIST for HCC patients getting radioembolization and chemoembolization and so on, because just shrinking of the tumor alone doesn't tell you the whole story. And so loss of viable tumor on imaging is a very important readout, which is how mRECIST works. But with immunotherapy, you have the added dimension of causing significant inflammation, which may be a very good thing for the patient. But when you look at that on imaging, the tumors may expand or swell. And so if you do imaging at 6 weeks or 8 weeks early, you may see what the radiologist will call progression. But if you wait and you re-image the patient 3 months, 6 months later, then you may see the response actually start to evolve. So I think it's very important that as a community, the community involved in developing therapeutics for liver cancer patients and supporting liver cancer patients, we need to make sure everybody's educated, including patients, to understand that the response to therapy may be assessed a little bit differently so patients can advocate for themselves, so the physicians caring for the patients understand it fully, that it may be worth staying in a clinical trial even if the initial report says progression. If the patient's doing well clinically, well, then wait, get another imaging study in 6 weeks or 8 weeks and see if the response is evolving. So I think the short answer is, we need to work together to create much more intensive and robust educational programs so that patients, physicians, regulators, politicians, lawmakers understand that this is an area in need of improvement. We need to support innovation, especially in the area of drug development and regulatory assessments and approvals, so that we can do liver immunotherapy development in a very rational way that aligns with how these drugs actually work.
Thanks, Dr. Katz. I really love that you just spoke about the patient advocacy and patient conversation piece. I think that's so important to guide the work that we do at Global Liver Institute. And I'm curious, what suggestions can you give on how patients can be guided to ask questions about immunotherapy and new treatment innovations?
When I talk to patients and when I've taken care of patients in phase 1 trials, I've really encouraged them to look at their reports. I think a lot of patients do this today anyway, as many have access to their electronic portals, but really helping them understand and educating them how to look at the reports, particularly the imaging or radiology reports, so that they can ask their physicians, what does this mean? It says progression, it says stable disease. Is it really a problem for me now? I feel great. I'm not having any side effects. My energy level is great. I feel like I'm doing well. Can I stay in this clinical trial longer to see how things evolve? I mean, that's the type of question that I think can be very productive, for patients to ask their physicians. And even outside of a clinical trial, patients who are getting checkpoint inhibitors for hepatocellular carcinoma, I think it's important to have that perspective, that knowledge, and the ability to ask those questions.
Lastly, Dr. Katz, if cell therapy succeeds in liver cancer patients, will it replace other forms of treatment?
I think the short answer to that is not necessarily. I think the frequency with which we use some therapies may change, the sequence or order may change, but I think things like surgical resection, checkpoint inhibitors, even drugs like sorafenib, lenvatinib, liver transplantation, and so on and so forth, TACE and TARA, these are all things that are going to, I think, remain part of the liver cancer treatment landscape. But cell therapy, if and when it succeeds for the treatment of patients with primary and metastatic liver tumors, is going to offer many more patients, we hope, curative long-term disease control options. And then when you have other tools in your belt, if you will, to combine with cell therapy or use in sequence, that's just going to offer liver cancer patients much bigger wins.
Thank you all for joining today's session and being part of the October is for Livers campaign. A deep thank you to Dr. Stephen Katz for sharing his expertise on T-cell therapies and their potential to transform care for patients with advanced liver cancer. You can find out more information about Global Liver Institute and the work we do at globalliver.org or explore livercentral.org, our hub for liver cancer research and resources. Once again, thank you, Dr. Katz, and thank you to our sponsors. Have a great day.